Hi everyone, how's it going? I hope you all are doing well today. It's beautiful weather on the East Coast where I'm at here in Maryland. My name is Shannon Farmer. I'm the NAPPC manager, so I probably interacted with you all in one way or another at some point. We're so excited to welcome you to our second annual Grantee, our Honeybee Health Grantee Reporting Symposium. We've been doing this Honeybee Health Grant for the past about 17 years or so. We've been doing this since 2008. So we're so happy that we're able to continue this and all these partnerships for all this time. But I'm going to go ahead and introduce also my colleague here, Reed Livers, who is our coordinator for a lot of our different programs that we have here at Pollinator Partnership. He is going to be our technical assistant today, and he's going to be looking over those Q&A channels, the chat, everything like that. And I'm also going to have him pull up our slides so that you all can see and share his screen. But I'm so glad that you all are able to join us today. First and foremost, wanted to give an acknowledgement of all of our different task forces that have been involved. Our Honeybee Health Task Force specifically has been a huge major part of this process in helping out choose all of our wonderful researchers that we have every year and kind of managing this entire grant. I also wanted to send a shout out to our task force co-chairs who kind of help manage the task forces and all of the tasks that come from that every single year. And we wanted to especially give a big shout out. Oh, there we go. Now our screen is being shared. It's going to take a second. Give a big shout out to our sponsors as well. We've had a few different sponsors this year, including our USDA APHIS grant folks who are from the Animal Plant Health Inspection Service, Valmont, as well as the J.M. Smucker Company. And of course, our program is run by the North American Pollinator Protection Campaign and our parent program, Pollinator Partnership. So thank you all for all of your support. This obviously has been a really challenging year for a lot of different folks. So we're so glad that we're able to really continue this Honeybee Health Grant because obviously honeybees are incredibly important to our agriculture, our economy, our very lives, which is incredibly important. So as we talk today, we're going to be able to talk about all of these wonderful researchers and all of their different projects and how they have helped out with honeybees over time. If we go forward to the next slide, you'll be able to see kind of the Zoom logistics. We're going to be in webinar mode this entire time. So for the most part, the only folks whose faces you'll be able to see is whoever is presenting at the time. So everyone's audio should be on silent. You shouldn't be able to speak except for the presenter. But you'll be able to see the audio settings on the lower left side of the control panel. Located at the bottom of your screen, you can click on the Q&A and type a question to any presenter. Our Q&A sessions will be kind of joint Q&A sessions with multiple of our presenters at one time, and you'll be able to ask that. So if you have a question while someone's presenting, go ahead and type it up. We'll bring it up during our actual session. Make sure that you actually reference who you have a question for. So if it's for Dr. Amiri who's presenting, Sagar, or Marie or anyone who you have, make sure that you actually specify who you're asking. If you have any technical difficulties, again, Reed will be monitoring that chat function. You can also email the NAPPC at pollinator.org. If you have any questions, I'll be monitoring that email. Reed will be monitoring that email during our presentation today. And then if we go forward to the next slide, you'll actually be able to see our agenda. So as folks are getting ready to kind of get started, I will be going over each person. I'll quickly show my face and say, okay, here's who's going to be going next. They'll present for about 10 minutes, and then we'll go ahead to the next person. We should be having a break or a Q&A session around 11:15 or so, and that'll be about 10-15 minutes, and then we'll go on our break, and then we'll come back and get started with the next set of our presenters. So for now, it is my honor to present Sagar Bhandari, who is going to be presenting his research, the impact of, oh, it went away, but that's okay. Sagar is going to go ahead and share his screen and come on screen with me. Hi, Sagar, perfect. Go ahead and get started whenever you're ready.
Is this screen visible?
Looks good.
Okay. Hi, everyone. This is Sagar Bhandari. I'm a graduate student at Mississippi State University in Dr. Amiri's lab. During Q&A session, I and Dr. Amiri will be addressing any questions you may have. So today, I'm going to discuss about the impact of viral infection on honeybee queen reproduction quality. Here is a presentation outline. Among several stressors affecting honeybees, viruses are the major concern. Virus can impact queen health, pheromone profile, and lifespan. Among several identified viruses in honeybees, deforming virus B is one of the most prevalent viruses. It is increasingly replacing other deforming virus strains and is also associated with colony loss. However, there is limited information on how this deforming virus B impact reproductive parameters of queen and its transgenerational effect under natural viral infection. Understanding this impact is key to improve queen health and overall colony performance. This figure shows different routes through which a honeybee queen can receive and transmit viruses. She can transmit vertically through eggs. She can receive viruses from nurse bees through royal jelly, and she can also be infected during mating. However, this study is primarily focused on the vertical transmission. We would like to understand how this deforming virus B impact several reproductive parameters of queen and how an infected queen transmit viruses to both worker and the drone-destined eggs. And except viruses, what else it transmits, particularly regarding immunogens. To understand this systematically, we set three objectives. First is to investigate the infection patterns of this virus and the immune response in queens and the eggs. It will have to understand whether the viral load and immune response is tissue specific. And that is to understand the impact on reproductive and physical parameters, and then the transgenerational effect of this deforming virus B infection in honeybees. Methodology. Around 50 queens in our apiaries were screened to assess deforming virus B infection status based on the viral titers in their eggs. 10 queens with low infection level having virus load less than or equal to 4 and 10 queens with high infection level having deforming virus B load greater than 4 were selected. Queens from each experimental groups were caged with worker and the drone frames overnight to collect worker and drone destined eggs respectively. After egg collection, queens were weighed and dissected to measure ovary weight and quantify virus load in individual body parts. We also took data for some physical and the reproductive parameters, including queen weight, ovary weight, egg size, and number. We measured egg size using ImageJ software. For molecular assay, RNA was extracted, and then we synthesized cDNA, and we conducted qPCR for the absolute quantification of deforming virus B and the relative quantification of 12 immunogenes. And we analyzed our data in RStudio using the best fitting models. Results and discussion. This figure represents infection patterns in low infected queens. The horizontal axis shows different body parts of queens and also the workers and the drone eggs laid by these low infected queens. And the vertical axis shows the virus load or the deforming B virus load. And it is clear that the ovary exhibited lower virus load as compared to other body parts. And in the case of high infected group also, ovary exhibited lower virus load as compared to head, thorax, and rest body. And again, we found no differences in the egg sample between worker and the drone eggs. Regarding the impacts on the reproductive and physical parameters, we measured queen weight and found no differences between low and high infected queens. However, the ovary weight was lower in the highly infected queen group as compared to the low infected queens. Though the ovary weight was lower in highly infected queens, we found no differences in egg numbers for those workers and the drone eggs. And again, there was no difference in worker egg size between the experimental groups. However, the size of drone eggs laid by highly infected queens was lower as compared to the eggs laid by the low infected queens. Regarding the immunogen expression, this dicer-like and Argonaute represents RNAi pathway, and both of these genes showed the similar expression patterns. We can observe differences in hair, digestive, and the spermathecae for both of these genes, with higher expression in the highly infected queens, and no difference was found in ovary and the egg samples. This defensin 1 is an antimicrobial peptide, and we found differences only in the head with higher expression in the highly infected queens. This avacin and epidacin both are antimicrobial peptide, and again we found differences only in the head with higher expression in the highly infected queen, and no differences in other body parts and the eggs. In case of this hymenotocin, which is another antimicrobial peptide, we observe differences in head in all the body parts with higher expression in highly infected queen, but no differences in the eggs. And if we noticed statistical differences, then every time the expression was higher in the highly infected queen group. In case of this release also, we observe differences only in the head. But in case of TOL6, we observe differences in workers and the drone eggs with higher expression or the immunoresponse in the eggs laid by the highly infected queens. In case of STAT, we observe differences only in the worker eggs. However, we did not notice any differences in the heat-shock protein, this B antiviral protein, and for the vitellogenin. Conclusions. As we notice lower virus load in ovary as compared to other body parts, we can conclude that ovary is the least infected tissue against deforming virus B infection. The virus exerts a targeted influence on reproductive quality. The findings on viral load, its impact on the reproductive parameters, and the immunogen expressions are consistent with each other. In relation to the viral load distribution or the infection pattern, ovary exhibited lower virus load comparative to other body parts or tissues, which might suggest less pathological effects in ovarian tissue. That is why there is limited impacts on the reproductive parameters. Only the drone egg size was affected. And regarding the expression of immune genes, no difference was observed in the ovary between the experimental groups except for one gene, which suggests subtle transcriptional differences in ovarian tissues. And again, we noticed differences only for a few genes in eggs, and it indicates that limited transgenerational effects under natural virus infection or the deforming B virus infection, at least through the immunopathways that we tested here. So at last, I would like to acknowledge funding agency, Pollinator Partnership, Project EpiSam, my lab members, graduate committee, and thank you for your attention.
Perfect. Thank you so much, Sagar. That was wonderful. We're going to hold any questions that we have for you until later on. So you can go ahead and go off screen for now. And if you folks have any questions, make sure you type those up in the Q&A and make sure that you acknowledge it that it is supposed to go towards Sagar. And for now, we're actually going to call up Marie next, Marie Calderon Rodriguez. She's going to be talking about her project. She'll be able to share her screen, and you can come on and start presenting whenever you're ready.
Yeah,
Perfect.
So just to make sure you can see my screen, right?
Looks good.
Okay. Okay.
Perfect.
So, okay. Well, thank you for the presentation and the opportunity to present our project here in this event. So our project is called Deformed Virus and Varroa Structure Dynamics in Puerto Rican Honeybees. So why do we study DWV and Varroa in Puerto Rico? Well, because our bees have some unique traits as mite resistant and they are less aggressive. So here we have seen that our colonies do not have not been affected as much as in the US and other locations. And our honeybees don't show the common DWV phenotypic characteristics in comparison to other places. So we determined the DWV prevalence, viral load, and viral destructor levels in our bees in managed colonies. And our hypothesis is that DWV is less viral in Puerto Rico due to stable and low varroa mite levels. So we collect samples from 3 locations, 2 commercial apiaries and 1 research apiary, the apiary that we use for our project in our lab. And our process, first we analyze brain transcriptome data from some previous project. And then to measure varroa and to measure the load, we sample bees from the 3 locations that I already mentioned. We storage the bees in -80. We perform RNA extraction, cDNA synthesis, qPCR, and then we choose some samples for PCR Sagar sequencing, and then we analyze the graphs and the t-test and everything with GNP probe. And for the varroa, we collect from 300 to 400 varroa from each colony. And we, for some projects in the future, we storage those varroa. So our findings, we found that we have DWV in both brain samples and whole body samples. Also, we didn't see any twisted wings or small bodies or dead bees for DWV, so that's a good sign. For next, we measure the viral load with both relative values and absolute concentration values. So we found the WV in our 3 locations from Gurabo, Macao, and Yauco. And we measured the copies per bee only to Macao and Yauco, and we found that our load is lower in comparison to Hawaii and the US. And for the varroa destructor infestation rate, we obtain an average of 3.82%, and we use the 4% to determine the high levels and low levels of varroa. So we obtain 15 colonies with low levels and 12 colonies with high levels. And we also use the colonies that we had results for DWV and varroa, and we obtain no strong correlation between the DWV load and varroa load. So to see which type of DWV is the most prevalent in Puerto Rico, we use 10 samples of the transcriptome data, and we found that all 10 samples were positive for DWVA, and only 2 of them were positive for both A and B. So we can see that here in Puerto Rico, we can see bees with only 1 type of DWV, and we can see bees with 2 types of DWV. So for conclusions, we obtain low DWV titers and multiple virus types, and we believe that support the DWV low virulence in our hypothesis. We suggest that our bees tend to have less virulent DWV, not only because of the mite resistance, but also maybe because our bees has evolved different the virus have evolved different in Puerto Rico. And our study is the first study to assess the relation between the WV low and varroa low in managed colonies. So for a future application, we will determine the relation between the DWV low diversity and varroa low across feral and domestic sites over time. And we want also to assess different manipulation treatment to see if our bees are resistant to DWV. So for the conservation implication, for the Puerto Rican honeybee population presents a really unique and awesome opportunity to study the potential benefits of targeting viral infections in a close uniform population. And our research reinforces the need to study viral and vector resistance to eradicate emerging diseases. So for the acknowledgement, I want to thank all my mentors and all the people and laboratories that helped me with this project. And also I want to thank Coordinator Partnership for the opportunity with the grant and to present this project here. So thank you.
Perfect. Thank you so much, Marie. If you folks have any questions for Marie's presentation, make sure that you go ahead in that Q&A function. Make sure that you address the question specifically to her so we know who you're asking about when it gets time for the Q&A. For now, we're going to bring up Sarah Red-Laird, who's going to go over her project. I'm going to hop on off because you seem like you're ready to go, Sarah, and make sure that you all monitor the chat if you have any questions for us.
Okay. Can you see the presentation all right?
Looks great. Yep.
And you can hear me?
Sound great.
Awesome. Okay, thanks. Let me just minimize this. All right. I actually am not directly affiliated with the university. I work for a nonprofit. And our mission is to create a world where bees enrich our working landscapes and our lives. The nonprofit is Bee Regenerative, although a lot of you folks might know me by my nickname of Bee Girl. So for this project, I was working out on a bison ranch in South Dakota that has been running a herd of bison for over 30 years. And the goals of the project is to find potentially mutual benefit between bees and bison. We've set up transects to explore bee nesting behavior, bee habitat, and bee communities. We're looking to discover flowers at the bison ranch that are both attractive to bees and high nutrient density through pollen, and how the nutrient density at the ranch compares to other ag landscapes across the West. This is a collaborative project with Vanessa at the Tucson Bee Lab. We're trying to understand the landscape management practices at the Bison Ranch. It's in transition, and so the current name is quote unquote the Bison Ranch of ownership. How they may influence bee communities and if found to be positive, how these methods could be replicated. We're also monitoring soil health and monitoring honeybee health and working to understand the relationship with the ranch's native bee population. So our hoped for outcomes are to collaborate with methods to increase flower diversity at the Bison Ranch and potentially other bison and cattle ranches, to educate kids and consumers on the cultural, nutritional, ecological, and tribal importances of bison as wildlife, as well as ranch-raised bison, to continue to share our findings with the scientific and ag communities, and to potentially utilize this data to advocate for the restoration of American bison onto public and private lands in the U.S. Just a note, this is a multi-year study. We plan for it to go at least 5 to 10 years. This is just the first couple of years, and so we're currently casting a really wide net and looking at a lot of different data in a lot of different ways to just kind of see what interesting or weird or cool trends are going on out here between bees and bison. So just to orient us a little bit, that is our study plot down there under those blue dots. The ranch itself is broken up into 20 paddocks. It's 28,000 acres altogether, managed using adaptive holistic plan grazing. There is about 1,200 honeybee colonies spread out over 17 bee yards, living alongside 2,400 bison, which means approximately 1.4 million pounds of bison, which are constantly moving through the landscape. So I have 7 transects set up all over the ranch in a lot of different ecological kind of sub-ecologies throughout the ranch. So we're looking at a pond area, pond and riparian area, area that has a bunch of wallows, bison wallows in it, 2 different rangeland areas with buttes and flats, a perennial swale, a perennial creek and riparian area, and then a control area with bison that are excluded and have been excluded. This is about a 11-acre pasture that the boys excluded the bison for me starting in 2022. So we're looking at bees through a myriad of ways. One is blue vein traps. Each study plot has 1 blue vein trap that's left for 24 hours. You're like, is your dog smiling in that picture? She is. This is how we're looking at vegetation. 25-meter vegetation transect with a square thrown out multiple times randomly. These are my this is an example of my floral host observation transect. We're also looking at nesting behavior observations and how those, how bison interact with ground nesting bees. This is our nutrient density study that we're doing with Vanessa, where I'm collecting different kinds of flowers from the ranch, but we're just focused on sunflowers right now, Helianthus Bhandari. Soil monitoring and honeybee health monitoring. Also, grazing behavior observations. And the honeybee health monitoring is done throughout the apiaries outside of my study plots. And then grazing behavior observations are also done outside of my plots, basically anywhere where I can find bees. Here's a little bit of a sampling of our data. These are the area with the wallows. As you can see, Apis mellifera look to be dominant here, but really it's because there was quite a bit of alfalfa flowers, and that the honeybees were pretty much covering the alfalfa flowers, and I only found 1 bee visiting 1 native flower, which was the white prairie clover. Moving into the riparian zone, this area had the highest amount of both native flowers as well as the highest amount of native bees. And again, you can see there were Apis mellifera present, but they really didn't co-pollinate on the same flowers at the same time. This is our range plot. Again, same trend as I mentioned before. This is our control plot with no bison, which is a really fascinating place to watch bees in these interactions. Look at all those honeybees out there. Really just 1 native bee spotted the whole entire season. And she was kind of all over the flowers because really alfalfa, yellow sweet clover, and sand lucerne were mostly dominant, which is what the Apis mellifera, of course, were all over. And then looking at this all together, at first glance, you think, oh my gosh, there's just really so many honeybees out here, which there really are. But I think it's fascinating that the honeybees really truly are pretty much sticking to their own flowers, whereas the native bees are sticking to their own. Bison are not interested in eating the native flowers. They pretty much avoid them. However, they love alfalfa and sweet clover and sand lucerne. And so what I've seen out at the control plot, where the bison are excluded, is the yellow sweet clover and the alfalfa are really out of control because the bison aren't there managing them. Whereas other places on the ranch where the bison are nibbling these flowers, it's really making more space for the native flowers to grow and thrive, and the native bees as well. I think the 2024 data is going to show some differences in trends because there was some climate change chaos out on the ranch last summer, which basically bees, and around the ranch as well. The alfalfa and the sweet clover were really scarce, and I did see a lot more competition. Well, I don't know if I would call it competition. There's a lot of co-pollination on some of the native flowers. This is our nutrient analysis. So far, we're not really seeing anything too out of the ordinary. These protein and lipid ratios are pretty much match the average of what you see in sunflower protein and lipids anywhere else, even on commercial farms. It was a little interesting how some of the plots really did differ, however, Old Snag. And they're looking at the landscapes themselves. All three of the plots that I took the sunflowers for nutrient density, the one that had the least amount of protein and lipids was the most heavily dominated by just this one flower, where the one that had the highest amounts of protein and lipids and the nutrients, the flowers were actually in a plot that had multiple other flowers blooming at the same time, and there was way higher diversity. So I thought that was really interesting. Here's just a sample of the soil enzymes that we're looking at. I thought it was also really interesting that in the control plot, the enzymatic activity was way down compared to the enzymatic activity where the bison were actively grazing. This is a video of bison. I know I'm running to the end of my time, but this is an interesting video that shows bison completely avoiding eating. There's curly cup gumweed and Bolander sunflower down there. And the bison are weaving around and they lick the grass or the plants before they eat it. And so they will munch the grass. They're not touching anything with a flower on it, which makes them a really great animal to live alongside bees, because they go along these fields of flowers, and once a sunflower has gone to seed, they'll nip the top of the plant off, and then they'll leave the flower for their bee friends. This is what's coming up next for us. Just a lot of cool education. We're also looking to expand, or we are expanding the project into 2 different ranches in Montana this season and next season, because I'm really interested. Bison are quite bee friendly. We're also really interested to know about look at the different interactions between honeybees and native bees, is what we're seeing on this bison and the relationship that they have with bees. So we're looking to replicate the study elsewhere. That's the biggest grasshopper I've ever seen in my whole entire life out in my control plot. And thank you so much for NAPPC for helping to fund this project, and for our crew at Bee Regenerative and the ranch crew for everything that, all the support that they've given and that you've given. Thank you for listening.
Perfect. Thank you so much, Sarah. If you folks have any questions for Sarah, just make sure that you go ahead and write that in our Q&A section. Make sure you address it to Sarah for our Q&A session. For now, thank you so much, Sarah. We're going to go ahead to Yosef Hambatola, who's going to present his presentation, and I'm going to hop off and take it away whenever you're ready, Yosef.
Can you see my screen? Sorry.
Yes, we can.
Okay. Thank you. My name is Joseph. I'm a postdoctoral fellow at in North Carolina State University. I'm presenting on our project artificial selection of the gut microbiome to investigate their impact on honeybee longevity and microbiome dynamics. Here I particularly focus on the workers' honeybees. From the workers' honeybees, the 9 core bacteria species have been reported so far. These bacteria are simple, they are simple and conserved, and it's easy to manipulate them for the experimental setup. Each of the bacteria, they do have the specific roles. They have different roles in the bee biology for digestion, nutrition, and metabolism. And they also help in protection against pathogens and neurophysiological functions. And they also play a role to modulate the foraging behavior. So in our current experiment, we focus on the microbial selection from the different lifespan and from different lifespan and the glyphosate exposed honeybees. We predict that inoculating newly emerged worker bees with a gut microbiota from short-lived, long-lived, and glyphosate-exposed donors will lead to difference in host lifespan and microbial composition. It is independent of the host influences. To conduct this experiment, our experimental design for microbial selection is the first step is collecting the mature pupae to get the newly emerged bees, and also collecting the forager workers from the hive entrance to get the gut homogenate for the inoculation in our experiment. And the second stage in longitudinal rearing in our selection, the one you can see here is a bee rearing cage design. The number you can see here is 1 to 6 shows that we conducted the experiment for the 6th generation. In the first generation, we had 3 distinctive treatments. Our treatment in the first generation is the first black one is control, and the green one is a mother for the short-lived and long-lived one. And the green one is, and the red one, sorry, the red one is a glyphosate treatments. For this short-lived and long-lived selection, after the gut colonization, the 50 day, the first 5 the short-lived bees, we treated them as the short-lived treatment, and also the last 5 long-lived bees, we consider them as the long-lived bees. So across this experiment, for each treatment, we had 5 cages, and in each cage 65 bees through the generation. And also we use the same sampling design across the generation. From this one, we recorded the lifespan, and also we did a molecular analysis for the microbiome. We did the dissection, the gut dissection, DNA extraction, and sequencing. After that one, the sequencing we analyzed using QIAM and also R language. From this analysis, our first finding is bacterial abundance remains stable across 6 generations. From this figure, as you can see, we grouped it by treatment across the generation. The first one is control from 1 to 6 generation. Second one is short-lived. Third one is long-lived. And last one is the glyphosate. And I can see that you can see the 50% below composition. I visualized here by removing the dominant taxa. From this one, we only find one bacteria, the one I underlined, Bifidobacterium asteroides, which showed the variation in one treatment, the glyphosate treatment, particularly in the 2nd generation. I will talk about this bacteria in the next slide. To build up on this one, we also analyzed the relative abundance in inoculum type to see if it predicts for the relative abundance over time. Here, the inoculum type did not predict the relative abundance over time. Not only the abundance, the experimental treatment also does not affect the bacterial diversity across the 6th generation. However, you can see here from this one, I showed only one matrix here. I did different metrics. In the last generation, you can see that in the long-lived had higher diversity compared to the short-lived one, but they are not significantly different. What we know is that reduced microbiome diversity in honeybee is linked to the short lifespan, and also the the gut microbiota disruption in insect impair for health and reduce longevity. Our second finding from this experiment is glyphosate exposure increase the abundance of bifidobacterial steroids in second generation. As you can see on figure A, the relative abundance of the bifidobacterium steroids in the second generation is significantly different compared to the other treatment. And also, if you can see figure B, we did the differential abundance of bifidobacterial steroids across the generation. If you can see from first generation to second generation, it's significantly increased. And if you can see from second generation to third generation, it's significantly reduced. And what we know about bifidobacterium steroid is it possesses the detoxifying properties that may counteract the glyphosate harmful effects. And also the bifidobacterium steroids has linked to the stimulation of the juvenile hormone, which regulate the insect growth, development, and longevity. In addition to that one, honeybees inoculated with the bifidobacterium steroids showed significantly higher foraging intensity. Our third finding from the experiment is microbiomes selected from the long-lived bees enhanced longevity. The survival curve you see from this one is the first generation to the sixth generation. Our selection, we started seeing the enhancement of the longevity from the long-lived treatment from third generation. If you can see the yellow curve, it is being separated from the other curves showing increasing the survival and through the sixth generation. I also supplemented this figure with the average extended lifespan. From the average extended lifespan from this figure, if you can see the yellow line is showing how the microbiome select from the long-lived is increasing the survival. So here it increased the bee lifespan by 5.2% on average across the sixth generation. Particularly if you looked at the fifth generation by 27% and sixth generation by 18%. From this microbial selection, our fourth finding is the microbiomes from glyphosate bees reduce the longevity. You can also see now from the first generation to the last sixth generation, the red curve is significantly reduce the survival. However, on the second generation, you can see that the glyphosate treatment showed the higher survival. That's why I put a question mark there. I can also connect this to the average extended lifespan, if you can see from there. If you recall from my earlier slide about bifidobacterium steroids, which showed higher abundance in the second generation, is correlated with this the higher survival in the glyphosate treatment. Here I'm not saying that the bifidobacterium increase the survival, but I'm just showing the correlation we found here. In general, the glyphosate reduced the bee lifespan by an average of 8.6% across the sixth generation. What we know about the glyphosate is that glyphosate disrupts the beneficial gut bacteria by causing the dysbiosis and weakening the host health. Here in our experiment, we found that the taxonomic stability persisted, but the physiological issues likely limited lifespan gains. The key finding from our experiment is that bacterial abundance and diversity remained stable over sixth generation. And the glyphosate exposure increased bifidobacterial steroid abundance in the second generation. And microbiomes from the long-lived bees extended bee lifespan by 5.2% average across six generations. Finally, the microbes from the glyphosate-exposed bees reduced the bee lifespan by 8.6% across the sixth generation. We also have an answered question from this experiment. The first unanswered question is regarding the functional adaptation. Can the experimental selection drive the functional adaptation in honeybee gut microbiome without changing its taxonomic composition? What we found in our experiment is a taxonomic composition which was stable across the generation, but we don't know about the functional adaptation. The second one is, does the strain level variation with the stable taxa explain resilience to the glyphosate and selection pressure? And the other one is about the bifidobacterium steroids. Is the longevity benefit linked to the bifidobacterium steroids under glyphosate exposure due to higher abundance or functional changes like altered gene expression? These are the few unanswered questions we raised after the experiment. Finally, I would like to acknowledge NRC for sponsoring my fellowship and also hosting University and NC State and for funding, and NAPPC for funding our project and apiculture program where I did my experiment, and Army Research Laboratory and other our collaborators, Ruben Lab, University of Alberta, and Raymond Lab from NC State. Thank you. I also acknowledge our agriculture lab members, and if you have any question, let me know. Thank you.
Perfect. Thank you so much, Joseph. So that was great. That was all of our presenters for our first half. So now we're actually going to go right into the Q&A portion. So our presenters from our first half, you can go on camera if you would like to. So I know Dr. Amiri will be answering for Sagar. You can feel free to join us, Sagar, Marie, Sarah, and then Joseph, if you all would like to join us. I know in the chat there was a couple of Q&A questions that a few of you were able to answer. I'm going to give everyone a chance to go ahead and type up some questions while you have that. And while you all are typing up some questions, I'm actually going to go back over some of these questions that we had and see if any of you wanted to expand on. So I know the first question was for Sagar and from Dewey saying you separated low and high infection levels on log 4. Did you do any further breeding to separate low and high infection lines, or was this just a separation on initial screening? Dr. Mary, I don't know if you wanted to expand on that at all.
Yeah, thanks so much. I did answer the question, but what I want to try to, what I'm trying to say is that this is part of a bigger project that we do here in Mississippi State University to understand when and how queens get infected with viruses and what's the consequences of viruses on the queen health and the next generation, mostly next generation. So, and then for breeding, yes, we do have a program or project that it's a follow-up project actually, that we select low and no infected the queens and then raise offspring from them and then select them for a couple of generations to see if we can make two lines of very highly infected or very low infected based on the egg infection. Yeah, we are following that idea.
Thank you so much. The next question, which it looks like Marie had answered, was about your presentation, Marie. You indicated that Puerto Rican bees are considered a closed uniform population. On what basis? Will there not be any further imports of bees, accidental or otherwise, into Puerto Rico? I don't know if you wanted to explain your answer a little bit or expand on that at all.
We consider Puerto Rico a closed uniform population because, well, we are an island and we don't expect exchange or trade of any bees. And even though our bees are a mix between European and Africanized honeybees, our gene pool have remained very stable over time. So that's why we don't expect any change in the future.
It looked like there was maybe going to be another question for you, but I'm not sure if the person got a chance to finish typing it. So I'm going to go on to the next one. This question was for Sarah. Is the sunflower Bhandari species a native? Do bison also leave the flowers alone in grazing?
Yeah, it is. Yep. Helianthus Bhandari is a native to the Midwest, but it grows really well out here on the West Coast as well. If it's planted, it doesn't usually just naturally occur. But it's that's the same flower that I have sent to Vanessa is the flower that I showed in the video that I showed of the bison just nipping off the tops as they went through the fields. And it was really interesting because that specific sect of the herd would pass through that gully that had those flowers blooming in it every day. And then because Helianthus Bhandari, Bullender sunflower, blooms from top to bottom, and then it goes to seed from top to bottom. And so the bison eat the plant and the seed pods, but only after the flower has gone to seed. And they'll eat it hours after it's gone to seed, but they sit there and they wait and they won't eat it if it's still blooming. And there are bees just absolutely covering it. And it was interesting because I didn't see a lot of honeybees on this plant. I was there in the same time in 2022, but with the loss because of the hailstorm and the heat and the wind and all the things, the loss of the sweet clover and the alfalfa on the property, I did see significantly more honeybees on the Bhandari this year. But another thing that Vanessa and I had been talking about is sunflowers are seen as a low nutrient food for bees because they have low protein compared to lipids. However, these are flowers that are native. They're absolutely swarming with bees. So something is going on here the bees like. And we're thinking that it's really the lipids that they need more than the protein going into winter. And so that might be why these flowers are really highly attractive, is that they did have significantly higher lipids than protein. So, and this was in August, so end of August, beginning of September. So definitely a late summer bloomer. But yeah, really interesting interaction between the bison and the flowers. Whereas cattle, I've observed, will just absolutely annihilate these flowers if they're hungry and they have the chance. But bison do not.
That's interesting. So I just sent something in the chat to you all just to see if anyone has any questions for any of our presenters. I don't believe Yosef had any questions yet, so if anyone has any questions for Yosef, please feel free. I'll give everyone a moment to go ahead and type that up. And then, of course, as always, if you all are kind of thinking about these presentations as you're kind of thinking of questions or going through this, you can always send a follow-up email later to the nappc@pollinator.org address. I'll actually put it in the chat for everyone to see. Just in case you think of a question later or want to get in contact with any of the collaborators, you can email that email, and I'll be sure to pass that along to any of our presenters as time goes on. Doesn't look like there's any open questions for now. I also want to give the chance for the panelists. Do any of you have any questions for each other? Any thoughts? Reed, did you hop on because you had a question?
Yeah, I did have a quick question for Yosef, so maybe I'll just hop in with that. I was just wondering, Yosef, about any conservation implications, any on the ground applications that you think beekeepers and folks like us here at a Pollinator Partnership should take away from the research that you've done. Just no worries if you don't have a clear answer, but just wanted to see.
Yeah, that's a great question. Thank you. In general, this is the microbiome, the symbiotic microbes associated with the honeybee gut microbiome. It's kind of a recent studies, but now it's kind of a hot area. Many people are working in different ways. The long-term plan is for the probiotics. But at this point, we are understanding the basic science, how this kind of different stressors are influencing. Like, for example, here we used one environmental stressors like glyphosate, which is very common, and also we have seen a lot of news about it, how it is affecting the honeybees and also how it is affecting the honeybee gut microbiome. Based on that one, we wanted to understand how this factor, the environmental stressors can influence the gut microbiome. Not only influencing by one exposure, we wanted to see it through across the generation. From generation 1, we did up to generation 6, and we tried to look at it, how it is affecting the gut micro, how it is affecting the survival. The take-home message for our beekeepers is that, considering what's happening in environment, especially relating with the glyphosate and also other pesticides, not only glyphosate, it's good to think about it whenever we are performing our beekeeping. That's a thing. And on the side of the gut microbiomes, still we are studying on these things to understand what they are contributing and how they are reacting to the environmental stressors. If they are kind of resisting or contributing something resisting to the environmental stressors, they are going to be helpful in the probiotic development. Thank you.
Thank you. That's very comprehensive. Perfect. Thanks.
Perfect. So if there's no more questions, I think we all can go ahead and head into our break a little bit early. So first of all, I want to thank all of our presenters who have already presented at this point. Thank you so much for your research, your contributions. It's been such an honor to be a part of this process, from the review process all the way up to now to presentations. Thank you so much for your work. If any of you in the audience, again, has any questions for them that you think about later, or maybe feel like it would be better as an email one-on-one, please do email that NAPPC at email that I put in the chat. And I'll be sure to pass that along to all of our presenters. But for now, we are going to go ahead and go on a break. We will come back at 11:38 Pacific time and 2:38 Eastern time. So just be sure to be back at that point. For now, we will see you all when we come back from break. Bye, everyone. So without further ado, I'm going to call up Dr. Chia Hua-Lin, who is going to be able to present on her research. She'll share her screen, and I will hop off so that she can go ahead and present.
Hi, good afternoon. Can you see my screen in the presentation mode?
It looks great. Yep.
Okay, perfect. Thank you. Hi, my name is Chia Hua-Lin. I'm a research scientist at the Ohio State University. And before I start talking about the project today, I want to give credit to the people, the hardworking people who has done a lot of work. And without them, I wouldn't have all the information to present today. So my undergraduate student, Allison Davis, she has done all the prep and sample collection and helped me really run the experiment smoothly. And Luke Heron was a technician back last summer and now a PhD student in Dr. Johnson's lab, Reed Johnson's lab. And he is one of the big masterminds behind the designing and executing of the experiment. And of course, my amazing students in Reed Johnson's lab. I just really want to give them the credit. So how did we start this project? This is my collaborator, Jim Jasinski, and back a few years ago, he was doing a biofumigation project with the mustard cover crop, Brassica jamsia. Back then, he was looking at the benefit of using this crop to control fungal diseases in pumpkin. Long story short, there was a drought and there was not really a lot of disease happen that to see a significant effect of the biofumigation. But what he has discovered is that the bees, honeybees, really, really love this crop. So when the mustard was in bloom, you could actually walk up to the field and just hear the field humming because of all the bees who just hummed through the bloom. And so we start to think about what we could do with this crop to help bees. It is very attractive to bees. It is fast growing. So the growers could actually time the planting to provide pollen and nectar resources for when bees need it most. So this could be a potentially attractive crop for growers as well, to provide both pollinator and soil benefits. So Jim and I did a pilot study to look at what honeybees are collecting during mustard bloom. And here I want to direct your attention to the fall crop. During the fall, our mustard crop, the brassica here, actually bloomed all the way through until the crop was killed by frost. And the honeybee colonies near the mustard plot actually collect pollen for until it was practically too late to be foraging, well into November. Whereas our control hive have very little collection of mustard, and they actually stopped collecting pollen early. Another reason we are interested in mustard was because these glucosinolates, it is a compound produced by the plant that have antimicrobial, antifungal properties, which is the reason for using this as a biofumigation crop. And a small study many years ago has found that the pollen actually mustard pollen contain glucosinolates. And we wonder if consuming mustard pollen could affect or mitigate the gut pathogens in honeybees. And in fact, there was a group of researchers in Europe that had found in laboratory settings when you feed glucosinolates to honeybees, you could reduce artificial Nosema infection. So we decided to do a mesocosm experiment to look at more of a colony level effect. So we built 12 of these cages at the research station here in Columbus, Ohio. And we aim to answer these two questions. First, will honeybees prefer mustard pollen over the typical Ohio fall pollen when they're given the choices? And the second question, will consumption of mustard pollen reduce Nosema infections? And so we had to do some troubleshooting to design with our feeder design. The bees didn't really care much about our pollen cups because it was, I don't know, at first we had a white one. And we added the yellow cup to the set, and we also strapped some honey sugar syrup to the post. And actually they so here's our bees on one end of the cage, and we have the feeder on the other end. And we did find that the bees somehow prefer the fluorescent yellow cup. So this is our final design. We have a little rain cover to keep the pollen from getting wet, and we have the yellow pollen feeder cup, a little syrup feeder to direct the bees to the feeder, and we have some tangle food to keep the ants from going up to our feeder. And so in each cage, we have a small honeybee colony with about 2 frames of brood and 2 cups. There's a yellow circle here. And we have 3 treatments. The positive control gets base pollen, that's the typical fall pollen, in both of these cups, so 5 grams each. And the positive control also get fumagillin in syrup as the Nosema medication. Oh, I'm sorry. The negative control gets base pollen in both cups and just simple syrup. And the mustard treatment group gets mustard pollen in 1 cup and base pollen in the other cup and simple syrup. And then we did a base Nosema spore count of each colony when we first moved them to the cage, and we kind of ranked them. And then we have this tier design that Luke came out with, I think it was genius, to assign those treatment. And the goal here is so we don't accidentally assign more of one treatment to higher or lower colonies with higher or lower Nosema levels. And so we did 3 blocks at 12 colonies total. 12 colonies each and 36 colonies total. And the pollen source came from a parallel studies. It was funded by Project APCM, Healthy Hives Initiatives. And I would not spend time going through the details of this project, but we collect pollen from these locations and other sites from the project. And Luke and Allison spend a lot of time sorting the pollen, and the little pile yellow pollen there, that's our mustard. So I want to show you the examples of what the bees did to the mustards. So on the left here, you see 1 cup, the lighter yellow, that's mustard pollen, and orange, that's the base full pollen. And on the right, that's the control with both base pollen. And 1 hour after these cups were set up, you can see how popular the mustard pollen was. And then 2.5 hours later, it was practically all gone. And we see this over and over again throughout our study. Now, when we look at the Nosema spore counts, and this is data from every individual hives, there's some interesting dips. And I want to mention in the second block here, we had to, because there was very, very few Nosema spores at the baseline. So we had to artificially give them some Nosema spores in the syrups to cause infection. But the lines were really like all over the place. And what we found was that fumagillin works really well. But we didn't really see a strong effect coming from the mustard treatment. And this is the data across all 36 hives. And again, at the end of the project, of each trial. And again, fumagillin significantly reduced the number of spores per bees, but we didn't see a strong difference between the control and the mustard treatment. So in conclusion, we found that honeybees prefer to collect mustard pollen before the base fall pollen. The mustard pollen treatment didn't show a significant effect on Nosema spore counts compared to the nectar controls. And we think this was possibly because the honeybees were not exposed to enough of the glucosinolates in our pollen, compared to the laboratory study done in Europe. And also there may be other stressors in those mesocosm designs because we were unable to sanitize, or we have to reuse those cages. So there may be some lingering effect as we switch the blocks. So lastly, I want to thank our research assistants and supports and collaborators, and our funding by Pollinator Partnership, Project APSAM, Healthy Hives, and the OPGMA. Thank you.
There we go. Perfect. Thank you so much, Dr. Chia. That was really great to see. Yeah. Perfect. So if anyone has any questions for Dr. Chia, please go ahead and use that Q&A function. Again, we're going to be coming back to those Q&A questions at the end of this session. For now, I am actually going to call up Dr. Alexander Walton, who's going to be able to present his presentation. And I'm going to go ahead and hop off. Feel free to share your screen whenever you're ready.
All right. Thank you. All right. I think you probably saw some of this already. Like I said, I'm going to present about the social immunity of the queen's retinue, and that this is a queen and her retinue. And I told you about the benefits of the retinue, but the potential dangers of the retinue could be transmitting viruses or other pathogens from the rest of the hive to the queen. And that would be very bad for the queen and for the hive if she were to be inundated with viral infections. So my question then is it possible that the retinue, rather than being a conduit for viruses and pathogens, is actually some sort of shield that can protect the queen from the diseases that may be present in the colony? And with that hypothesis in mind, my predictions are that the retinue bees will have a heightened immune response, and also that the infected bees that have viruses, will be less likely to join the retinue. And so I'm going to focus on the second part of the experiment, the second prediction. And in this first experiment, I did an experiment in the lab to ask whether viral infection could affect the attraction to the queen pheromone. So what I did was I took bees of a known age. All the bees in this experiment are controlled for age, by the way. I'll just get that out of the way now. But I took bees of a known age, and we inoculated them with virus, in this case, Israeli acute paralysis virus, using this method we developed in the Rueppel lab, where we spray them with sugar water that is inoculated with virus and kind of toss them around in a cup, and they end up licking it up and they get inoculated with virus. We've shown that this works to inoculate the bees with virus and cause mortality. Then I put them in cages, plexiglass cages in the lab, and I expose them to queen pheromone using a pseudo-queen strip, this piece of plastic infused with synthetic queen pheromone that beekeepers use. I merely tape this to a microscope slide it into the cage, and then I can measure retinue response using a retinue assay, where I count the number of bees that are actually antenating this microscope slide over a period of 15 minutes. And they kind of treat it like a queen almost, and form a retinue. You can see that in these photographs I've taken. These are of cages looking down into the cage, and there's a piece of pseudo-queen that is taped to a microscope slide. You can see on the left image, there's a lot of bees that are performing this retinue response. And on the right side, you can see there are fewer bees that are performing the retinue response. So there's variation in how many bees in a cage will actually respond to the queen pheromone. And so I had cages that were inoculated with IAPV and control cages where I just sprayed the bees with sugar water without virus, and I measured their response to queen pheromone for several days. Here are the results for that. You can see the red line here is cages that received a virus treatment. The blue line is cages that were the control treatment. And the x-axis is just days over time after inoculation. And the y-axis is the average proportion of bees in those cages that were responding to the queen pheromone, that were performing the retinue response to the pseudoqueen. And you can see for the first 4 days after inoculation, there's a significant difference between the two treatments, where bees that received viral inoculation have a lower response to queen pheromone than my control bees. They're less likely to join this artificial retinue. But this effect does disappear over time. You can see the lines start to converge, and that could be due to potentially the bees actually fighting off the virus, or any bees that were particularly sick with virus have died, and now the lines converge. So in this experimental setup, we're seeing an effect of viral treatment on retinue response to queen pheromone. But I also wanted to ask, in addition to doing this experimental manipulation in the lab, what about in just sort of in natural conditions? If I open up a hive and some bees are performing a retinue response, do those bees that are actually in a real retinue, do they have lower levels of virus circulating than other bees in the colony, control bees of the same age again? And so, I did this by going into colonies, grabbing age-matched bees that were part of the retinue. You can see these bees down at the bottom that are antenating the queen for at least 15 seconds as she passes through the colony. I sampled them as retinue bees. As control bees, I sampled bees of the same age that were on a brood frame that did not contain the queen. And then I also had this third group that I call queen avoiders. And this is a behavior that I haven't seen in the literature before, but I've spent years observing retinues, and this is something you start to notice when you stare at queen retinues. These are bees that will interact with the queen, encounter the queen, and then rapidly walk away from the queen and even run away. So here's an example. If you follow this bee that the arrow is pointing at, you can see she will approach the queen, encounter her, and then quickly back up and leave the area. So these are bees that are actively not joining the retinue. And I call those queen avoiders. And so I sampled just naturally occurring retinue bees, queen avoiders, and control bees, and asked whether they had different levels of virus, in their bodies. I'm going to show you the results of one virus of the several that we've been looking at. This is black queen cell virus, a virus that affects queen, but is also found in workers. And here we see that the bees that avoided queens had a higher level of black queen cell virus than bees that actively joined the retinue. The control bees neither group was significantly different from control bees, but avoider bees had a higher level of black queen cell virus than retinue bees. So in conclusion, I see evidence here that sick bees do avoid joining the retinue by looking at both an observational study and a survey observational study, and also an experimental manipulation study, where worker bees that were exposed to virus are less attracted to the queen pheromone, but also retinue bees that are naturally in a hive have lower virus titers, specifically black queen cell virus, than the queen avoiders. And so this can have important considerations for beekeepers, who perform pollination services, because they need to consider the retinue as potentially as a queen shield. And one particular context where this might matter is in shipping queens, where beekeepers will ship or import queens, and they're delivered in these cages, sometimes with attendant bees, sometimes without attendant worker bees. But when they do attendant bees, they're kind of randomly sampled nurse bees. And it may be important to actually preserve the queen retinue and grab bees that have a high response to queen pheromone, because they may have lower levels of virus. So that's something I think will be important to continue to think about in the future. And with that, I'd like to acknowledge NAPPC for funding this research, and the Rueppel Lab, specifically Brianna Bevan, who is an undergrad in the lab, who helped do a lot of this work. And with that, I will say thank you.
Helps if you can see me. There we go. All right. Thank you so much. Appreciate it. And thank you so much for your flexibility and kind of starting back over there. So if anyone has any questions for Dr. Alexander Walton, feel free to use that Q&A function. Again, we're going to be coming back towards the end of all of our folks to be able to do a Q&A session all together. So for now, we're going to keep on going. I'm going to call up Austin Martin, who's going to present his work. And right after that on deck is Dr. Kara Wagner. I'll hop off and let you get started, Austin.
Sure. All right. Just going to share my screen here. Can everybody see this okay?
Yep, we can see it.
Great, thanks. My name is Austin Martin. I'm a PhD candidate at the Temple University Department of Geography and Urban Studies. And so I want to note this, I'm in a human geography department. So primarily I'm a social scientist. And however, I do have a master's in ecology, and I have a background in particular with urban bee research, and doing my best to integrate the two. And because a subfield I'm in is political ecology, and there's an ongoing conversation in political ecology about where is the actual ecology in political ecology. And so as an urban political ecologist, I'm committed to actually working with biophysical data. And so to that end, this grant has provided me an opportunity to further an aspect of my doctoral research and kind of an offshoot of my doctoral research. And so I'm also a visiting assistant professor at Villanova University's Department of Geography and the Environment. So I'm kind of in two places at once right now. And this research in particular is focused on Philadelphia. Initially, I wanted to bring in research on native bees in both Philadelphia and Montreal and their impact on honeybee health. However, that research that data is still getting analyzed. This in particular is going to be a focus on a floral survey in Philadelphia and the implications for honeybee health for that floral survey. And if you'd like to reach out to me, my email is right here, aumartin@temple.edu. So overall, with this particular focus, I'm looking at 48 sites across the city of Philadelphia, and sampled from both invested and disinvested sites. Those invested sites include things like parks with green space investments and things like rain gardens. And then the disinvested sites are basically vacant lots. And Philadelphia, as a post-industrial city, has a lot of vacant lots. In each of these sites, I measured floral species richness, the percent coverage of the floral cover, and the evenness of the floral species community composition. And what I found was that overall disinvested sites provided comparable floral abundance and greater evenness. And I also found that this highlights the critical role that spontaneous urban vegetation can play for honeybee health. A bit of background here. Catherine Baldock and a number of other researchers have shown conclusively that cities, urban areas, can be actually a refuge, a vital refuge for pollinators across the board. This includes honeybees. And the honeybee is dependent on consistent seasonal forage across the season, including in the city. Green space investment shapes the floral landscape in cities, but it might not meet ecological needs entirely. And so my goal here is to understand these dynamics so we can understand how cities can be an important haven for honeybees. On the left, the visual on the left shows a comparison from Baldock's team showing between urban, farm, and nature reserve. The top is abundance and the bottom E is richness for bees. And anyway, so I'm also drawing from Doug Sponsler. He's actually on my PhD committee. In 2020, he did a really interesting study looking at 12 apiaries across the city of Philadelphia. And he sampled from the pollen, did a meta-barcoding analysis of the pollen down to the genus level, and found that basically was asking the bees what plants are growing across the urban landscape. And he found that key forage plants are often emerging in spontaneous vegetation. And according to his assay, the flowers, the floral species the bees most dependent on, the honeybees are most dependent on, are clovers, goldenrod, sweet clover, a number of others that we'll get into here. And so my research questions here are, how do invested versus disinvested sites in the city differ in floral richness, cover, and evenness? Which key forage plants appear in each landscape type? And how do these findings illustrate the sustainability fix dynamic within urban political ecology? The sustainability fix is a framework that political ecologists use to describe the way that cities will invest in sustainability and green spaces in order to advance property values, to raise property values, and in order to as a sort of branding mechanism for the city. But those investments might or might not have a beneficial ecological or sustainable impact. And so I want to use some of that critical social science theorizing from political ecology as a framework to guide how I'm asking these questions and collecting this data. And so, yeah, again, I collected from 48 sites across Philadelphia. I also have data from Montreal, but that's going to have to come later. And I sampled all across the city, West Philly, Southwest, North, Northwest, Northeast, even split both 24 invested, 24 disinvested. This was between July and September, early September, focusing on peak bloom season. On the bottom here, you see pictures of a vacant lot on the left, a rain garden, which represents an invested green space in the middle there. And on the right, that's an infrared remote sense, an image from Landsat. It's a remote sensed image, which can highlight the contrast between impervious land cover and vegetative land cover, which is red. And so I'll be using that also further as I go on here to analyze landscape, the effect of the landscape too. Anyway, so for my floral surveys, I recorded blooming plants for each site. I estimated the ground cover of the flowers, and I calculated the species richness and the pilus evenness. I'm not sure whether I'm pronouncing that correctly. And I also identified key bee benefit genera like Trifolium, Solidago, Taraxacum, etc. And so for my data analysis, I did descriptive statistics and t-tests. I visualized box plots of floral richness and evenness. I cross-referenced my floral data with the Sponsler study, and I focused on the difference between invested and disinvested spaces. I didn't find any meaningful statistical differences, but I did find that disinvested sites showed a slight trend toward higher evenness. Here's a visualization of floral species richness, which also shows not much of a meaningful difference between the two. However, when I started to identify and cross-reference some of the key forage species that Sponsler's study highlighted, the ones that the bees are most dependent on in the Philadelphia urban landscape, I did find that the disinvested sites most frequently hosted those key bee forage genera. That includes Trifolium or clover, which were occasionally found in invested sites, but very frequently found in disinvested sites. Solidago, which were rare in invested sites, but very frequent in disinvested sites. And Verbascum or Moline, I'm not sure if I'm pronouncing that correctly either. That's pictured here on the right. That was also, I just want to highlight that as a being frequently hosted in disinvested sites. And so lessons learned here were that spontaneous vegetation does provide critical seasonal forage, and investment-driven greening in Philly might prioritize aesthetics over ecological function in times. And so my findings also illustrate the dynamics associated with the sustainability fix as theorized in urban political ecology, this idea that Philly is using ecological and sustainable ideas as a form of boosterism and a way to raise property values, but it might not necessarily translate into meaningful ecological differences. And so what I'm hoping to move forward with is this idea that cities can actually serve as a vital refuge for bees if diverse, spontaneous floral resources are maintained in green space invested sites. What can we learn from vacant lots, essentially, is what I want to go forward with here. Challenges, adjustments. I had sometimes occasional field disruptions when I was sampling for native bees, which I'm going to integrate later, that data. My pan traps were sometimes discarded. Sometimes sites were mowed in between my sampling. And also, midway through this process, I lost my PhD funding. That came to an end. I had to pivot to my full-time visiting assistant professor position in order to get money over at Villanova, which is great. Here's a picture of me taking my Villanova students on a field trip, an urban beekeeping field trip. So I'm furthering this knowledge, at least in the classroom here. But also, my native bee specimen analysis is ongoing, and my floral survey analysis is going to be integrated into that going forward. So my future directions, I'm going to complete my native bee identification, going to integrate that, and I'm hoping to take the insights from this research and advocate for urban planning that values spontaneous and seasonal forage diversity, so our cities can be better havens for both honeybees and native bees. So I want to thank the Pollinator Partnership, the NAPPC, and also the Temple University Department of Geography, Environment, and Urban Studies. And again, if you want to reach out to me, my email is right here, aumartin@temple.edu. So thanks, everybody.
Perfect. Thank you so much, Austin. That was great. So if you all have any questions for Austin, make sure you hold on to those till our Q&A session. Make sure that you actually address it to Austin as well. Up next is Dr. Kara Wagner, who is going to get her presentation started. So I'll hop off and you can start whenever you're ready.
All right, thanks so much. I appreciate the chance to come and share our progress on development of a beekeeper-friendly assay to quantify grooming behavior. So grooming behavior is a social immune mechanism of honeybees that helps them defend against pests and diseases. This behavior, of course, involves the use of legs and mandibles to remove pests and debris from either the honeybee's own body or the body of a nestmate. And there are some existing methods to quantify grooming behavior, but these tend to be pretty labor intensive and can involve expensive equipment like microscopes. One of the main ways that grooming is quantified right now is by looking at mites under the microscope and looking specifically for chewing of mite legs, which is a sign that they have been groomed off of the adult honeybees. So our goal here was to develop a simple and inexpensive assay that uses equipment that beekeepers may already have on hand to quantify this grooming behavior. One thing that honeybee keepers typically have on hand, and if they don't, it's easy to get, is powdered sugar. So beekeepers often use powdered sugar to perform mite counts. It is known to induce grooming. If you coat the bees with it, they'll groom it off. Another piece of equipment that many beekeepers have is something called a refractometer. This is used by beekeepers to measure the sugar content of their honey so they know when their honey is concentrated enough to be bottled and it's high enough sugar content. So our idea here was to use the powdered sugar to induce grooming behavior and then develop a standardized method to collect that groomed sugar and then use the refractometer to quantify how much sugar was groomed, so quantify that grooming intensity of the colony. Our first objective was really to design the assay itself to determine the assay components. So we were looking at, what physical setup do we need, but also what's the quantity of sugar we need to coat, and what number of bees do we need to use, what's the water volume we need to use to dilute the sugar, and the assay time. And all of those combined need to achieve a distinguishable concentration of sugar for the low and high grooming colonies. And then, of course, we wanted to assess how the assay performed. And we were especially interested in looking at the relationships between the amount of sugar that was groomed off of that collection of bees and the relationship between the sugar groomed and both the genetic background of that colony and also the mite infestation levels. So I'm going to share data just from one apiary for the purpose of time and simplification. But at that apiary, we had 13 colonies. This was less than we'd hoped for, but we had some shipping losses and financial limitations. But we did have 13 colonies. 2 of those were high grooming breeder queens from Jeff Berta. Then we had 5 queens that were F1s of grooming breeders, and they were mated in the grooming yard before being sent to us in North Carolina. And then our third group, we had 6 queens. These were F1s of the grooming breeders that were mated. They were shipped as virgins and mated in our non-grooming apiary here in North Carolina. So we predicted a negative relationship between these 3 colony types and the amount of sugar groomed. In other words, we expected those breeder queens here to groom the most sugar because they were bred specifically for grooming. And then this group of colonies that had mated in our non-grooming apiary and were F1s of those groomers, we expected to groom the least amount of sugar. We also expected a negative correlation for the amount of sugar groomed and the mite loads. So we expected the colonies that groom the most sugar to have the lowest mite loads because they were, again, selected for grooming. We learned very quickly that it was actually not feasible to do this with a honey refractometer. So the quantity of the sugar that we needed to hit that scale of the honey refractometer, which is used to measure honey, so that Brix score, that Brix range is pretty high, up to 90% sugar concentration. The amount of sugar that we needed to hit that scale was so much that it actually obscured the light. This is probably because of the starch that's found in powdered sugar. But being a former brewer, I actually had a brewing refractometer laying around the house. And brewing refractometers do the exact same thing. They just operate on a lower scale. So the Brix scale for the brewing refractometer was 0 to 30% sucrose solution. But we know that most beekeepers, don't probably have a brewing refractometer laying around. So we came up with a new idea, and that was to actually mix the powdered sugar with some beet sugar so that our results could be evaluated with or with the refractometer. So beet sugar is very red. And so the idea was to develop a scale similar to a pH color scale where we could look at the grooming level just by the color of that output. So in terms of the assay design, we ended up using a 50 ml Eppendorf tube fitted with a cap that we cut a hole in and then fit with a metal mesh screen inside. We collected about 20 milliliters or 50 bees directly from a brood frame. And then added a 1/4 teaspoon of that mixture of powdered sugar and beet sugar. So you can see that right here on the top of the mesh. We would just push that down through the mesh. This was essentially the right amount of sugar to coat all of the bees, but to not have an excess of sugar in the tube. We would then invert the tube and allow grooming for 5 minutes, and we would collect that powdered sugar. We could actually weigh that powdered sugar directly, or we could transfer the sugar into one of these small centrifuge tubes and add a milliliter of water and then evaluate that liquid either by the color on that color scale or using that brewing refractometer. And so the low and high grooming levels kind of fell within that range of the brewing refractometer. We found that the weight of the sugar groomed was strongly correlated with the refractometer reading. This is, of course, what we would expect. You can tell from this this high R-square value that this is kind of leaning towards the chemistry side. We don't usually see this good of a relationship in ecology. But basically all this tells us is that both of these are reliable methods for measuring the assay outcome. You can weigh the sugar or you can dilute it with the water and use a refractometer. We did see the predicted trend for the effect of colony type on quantity of sugar groomed. It wasn't statistically significant, but I think this is probably a sample size issue. But you can see here those grooming breeders those breeder queens did groom the most amount of sugar. And the F1s that mated in our non-grooming yard groomed the least amount of sugar. It's also not too surprising that we see kind of high variation in these open mated queens compared to the breeder queens, but again, this only represents two queens, so that may also be playing a role. We found suggestive evidence that the mite infestation was actually positively correlated with grooming, so this is the opposite of what we predicted. We think it may be related to a priming effect of the mite infestation. So these assays were actually performed, these grooming assays were performed in August when the mite loads are probably relatively high in the colony. And we know that mite presence can actually induce grooming. So these bees may have just been more prone to grooming behavior if they came from a colony with higher mite loads. It may also have to do with hygienic genes from our North Carolina mating apiaries. So I do work a lot with hygienic bees, and we tried to target non-hygienic or low hygiene colonies, but it may be that these colonies had lower mite levels because they were more hygienic. So moving forward, we'd like to repeat this with low mite pressure situation, probably in the early spring with a larger sample size. And I think it'd be really interesting to compare the assay results directly with results from existing grooming assays. So comparing the sugar results with actual mite biting analyses. And then if the results from that are promising, we can continue to work on development of a collection device that just streamlines that collection process a little bit better. This, I think, is really important, not only for honeybees, but also for native bees. We know that honeybees and native bees share floral resources and that virus spillover from honeybees to native bees occurs at these floral resources. So I think anything we can do to improve breeding of bees that are better able to maintain low levels of varroa, low levels of disease, has a lot of potential to also benefit native bee health. So with that, thank you to NAPPC and Jeff Berta for supplying the queens, and I'm happy to take any questions at the break.
Perfect. Thank you so much, Dr. Kara. So that was perfect. And like Kara said, if you have any questions for her, feel free to make sure that you address them directly to her during that in that Q&A function at the bottom of the screen. Next, I'm going to bring up Dr. Sasha Niklish, who will be presenting his. And he will actually be our last presenter. Right after he's done, we'll be able to actually jump right into our Q&A session. And that's when you all can kind of have your questions at that point. For now, keep them in that Q&A function, or we can wait till we're live. So I'll hop off if you're ready, Sasha.
I am ready, but I think you blocked my video.
Oh, sorry.
No
Let
problem.
me bring you back.
Yeah.
There we go.
Oh, there we go.
You should have gotten it. Yep, perfect.
All right, everyone
Perfect.
can see me now. Great. All right, let me share my screen here. Hope everyone can see that. Okay, well, thanks. Good. Hello, everyone. My name is Sasha Niklish. I'm an assistant professor here at University of California, Davis, Department of Environmental Toxicology.
Sasha, not to interrupt you, but it's in the view where we can kind of see your notes and everything. So if you go to presenter view, there should be a way for you to swap that out.
Oh, that's okay. I have no secrets.
Just in case you do, thought I should let you know.
Okay, I'll try it again.
Okay.
Here we go. How's that?
That's good.
Good?
Perfect.
Okay, thank you so much. I appreciate it. Thanks again for allowing me to provide an update on our project here, which is identifying novel synergies to increase the efficacy of current use miticides for varroa treatment. So I'm by training, I'm working here in Department of Environmental Toxicology at UC Davis. My training is I'm a protein biochemist, so I'm not an entomologist. Bear with me with your questions, but that's also the reason why I'm pairing up with Dr. Chia Hua-Lin. She's a research entomologist here at USDAR's honeybee lab here on campus, and the main driver actually also of this project. And I want to start also with the acknowledgement, of course, of NAPPC and Pollinator Partnership and Project APC to support this research. before I get into the data, I just you need to allow me to have like 3 or 4 intro slides just to bring everyone on the same page. It might be a new concept, so I just want to be careful introducing you to this system. All right, so what are we doing in the lab? In general, we're interested on a macro scale on the different types of multi-chemical exposures that the bees experience, as you can see here. But then also like on a medium scale, where we like to understand better how do these chemicals get transferred across the different hive members and stored also in the different physical sinks in our hive. And then on a micro scale, we're very interested actually how bees, but also their pest organisms here, you can see Varroa, defend themselves against chemicals, toxic chemical accumulation. So we're specifically interested in these molecular defense proteins, transporters, or pumps. You can consider them as efflux pumps. And what they do is they just keep chemicals out of these organisms. So we're trying to better understand how do they work in bees for risk assessment, but also how do they work in varroa mites, maybe developing better treatment strategies here, because these are key regulators of chemical uptake. These defense proteins, as I mentioned, is one of the main focus, also what I'm talking about today. And this concept is not new. Actually, over 40 years, almost 50 years ago, they were discovered, these pumps in cancer cells. That's why we often refer to this as multi-drug resistance. These cancer cells, but also bacteria that you can see here, they possess these pumps. And what they do is they recognize any kind of drug or treatment and immediately pump them out of the cell so they cannot accumulate and be used as treatment. So in fact, FDA, as you can see here on the top left side, demands companies to screen all their drugs and chemotherapeutic agents before they bring them on the market to test them against these transporters, because they're so efficient actually in pumping out these chemicals. In recent years, we also understand that arthropods and insects use these transport systems, these pumps, to develop resistance against certain types of pesticides. So you can see a couple of articles. And that was actually like what sparked the interest in our lab, like understanding how do these pest organisms use these transporters to protect them, and can we somehow interrogate this? Can we work with these transporters specifically in 2 areas? So one of these areas that we're interested in is like trying to understand how chemicals interact with transporters in honeybees, maybe allowing us to predict chemical bioaccumulation in these bees. And then vice versa, we also would like to understand, can we target these efflux pumps, these transporters and pest organisms, to inhibit them selectively only in the pest organism, and to make possibly current use pesticides more effective. And this is actually something that I would like to talk about today. So this is the part of the introduction where I hope everyone is still with me. So what do we do? We have a 3-pronged approach here. I mentioned I'm a protein biochemist. I work also on structural biology. So given the recent advances in AI and machine learning, we are able actually to model proteins, including these efflux pumps. And what we're trying to do is we're screening a large amount of chemicals from different databases that you can see here, coconut, like natural compounds, but also drug bank, and in silico. So in our computer, we can screen several hundred thousands of chemicals, trying to understand, could they be an inhibitor or blocking these pumps in varroa mites? And then in a second step, we are using assays in our lab. This is an activity assay that you can see here, a medium throughput activity assay, where we can compare pretty much different inhibitors to each other. Do we have a strong inhibitor or a weak inhibitor for these efflux pumps and varroa mites? And then in the final step, of course, we want to understand and validate in vivo with a couple of compounds that we then prioritized. Okay, do they inhibit these efflux pumps and varroa mites? Are they toxic to these mites also? But the same compound ideally should not have an effect on honeybees, of course, in these assays. And this that this principle or this concept that this is possible is actually goes way back actually during my postdoc time, like trying to understand if we can have the same compound that is toxic to one organism, but less toxic or not toxic at all to the other organisms. I was a postdoc at Scripps Oceanography, and there actually tried to understand chemical accumulation in food organisms and fish. So here I worked with yellowfin tuna on these transporters and with mice. And I give you an example here. I did an assay for testing for these compounds that can block these efflux pumps. And here you can see in blue a typical inhibition curve of this assay. And you can see that the tuna transporter in blue and the mouse transporter in red have different potencies of inhibition. In fact, like I was able with the same compound to inhibit the mouse transporter 60% more than the tuna transporter, right? And that was actually the question that I had when we tried to translate this knowledge of these transporters from mammals, from vertebrates into invertebrates. Can we do the same for the bee, right? And for the varroa mite. So can we create an inhibitor? Can we find an inhibitor that is less toxic, less blocking for the honeybees, so that they can still use these transporters to defend themselves against chemicals, but it's more inhibitory for the varroa mite. Okay. Long story short, we did this. So we tried to, with the help of a postdoc in my lab, Jagan, we modeled these two proteins that I was talking about, these two efflux pumps from the honeybee and the varroa mite. You can see them here. We superimposed them. This is a model of these transporters for the honeybee and the transporter from the varroa mite. And then the first step that we did is like we looked at how similar are they? Is this, hopefully they are not similar enough. So we can actually start developing inhibitors that are precise and specific to the varroa mite. And we were lucky. So here on the top right-hand side, you can actually see almost only 23.4% of these transporters between the honeybee and the mite are similar. So the amino acids in these transporters. And if we look a little bit more in this, what I marked here in the rectangle, this is actually where do these pumps interact with chemicals, with pesticides, with miticides? You know? So this is the section of this protein. And if we look at that, we can see, oh yeah, they're even in the microstructure, these letters and numbers represent amino acids. We can see there's a large difference between the honeybee transporter and the varroa mite transporter. So that was our second check. So there is structural difference in these transporters that would allow us maybe to ask the question, can we find compounds that are strong inhibitors? They block varroa transporters, but they show weak or no interaction with the honeybee transporter. Okay. Again, long story short, they exist. So we did these molecular docking studies that I showed you in the beginning in the slide. And we used amitraz, arguably maybe one of the most efficient miticides on the market right now. And to our surprise, actually, we saw with these studies that amitraz is a substrate. Amitraz can be recognized as a substrate in varroa mites, so they can actually pump it out. So what happens if we add an inhibitor on top of a substrate? Does that maybe allow us to keep amitraz in the mites accumulating and cause higher toxicity? We identified an inhibitor here on the right-hand side. You can see verapamil that we identified as an inhibitor. And we wanted to use this inhibitor as a synergist to increase the toxicity of amitraz in varroa mites. And so with the help of a talented undergrad student, and of course with the generous support from NAPPC and Pollinator Partnership, we were able to obtain this really nice microapplication setup that you can see here on the left-hand side. It allowed us to calibrate nanoliter amounts, to inject nanoliter amounts topically onto our mites. You can see here on the wax foundation, and we calibrated, you know, like 150, 200 nanoliters of our solutions to be able to apply definite amounts of these compounds to the varroa mites. And here on the right-hand side, you can see what Julia mainly was the driver of these experiments. This is 4 replicates, always 16 to 20 mites that we treated. This is a 48-hour survival of control mites exposed to our inhibitor verapamil to amitraz. You can see here a reduction in the survival by 20-25%. But if we add our compound verapamil and amitraz together as a synergist, we can see a further 50% increase in mite mortality. And to our surprise, and with the help of our collaborator, Frank Rinkovich in Baton Rouge, we sent out these mites to him, and he came back genotyping them. We saw that almost all of these mites were showing the resistant genotype, the amitraz resistant genotype, meaning that we were able to use the synergist and increase the mortality or efficacy of amitraz by 50% in mites that are resistant to amitraz. Okay. So this is a study that we just submitted to Journal of Apicultural Research. It's still in review. And of course, we would like to find other additional compounds that we could use as synergists in the future now. And then the next step would be, of course, also to test this in the field. Yes, with this, I thank you and NAPPC and the Pollinator Partnership. Of course, I'm open to questions later in the Q&A.
Perfect. And you are actually our last presenter, so we are actually ready to go right into the Q&A session. So if you want to stop sharing your screen, and I'll have the presenters from the second half, so Dr. Chia Hua-Lin, Dr. Alexander Walton, Austin Martin, Dr. Kara Wagner, if you don't mind sharing your screens, goodness, sharing your wonderful faces is what I meant. And let's go to the Q&A. It looks like you all were pretty busy with some questions during the time, and I don't see any new ones right now. So I'll give the audience a moment to go ahead and type in your questions. For now, I'll kind of read over the questions that were answered. And if you as the presenter would like to expand on it, you're more than welcome to. So looks like this first question was for Dr. Walton from Dewey Karen. And he had asked, is there or will there be a quick test to determine bee viral loads to seek to the proper bees for the queen shield? Interesting that you found a new bee behavior in the queen avoiders. I don't know if you wanted to respond to that at all more than what you had already said, Dr. Alexander Walton.
Yeah, I'll just reiterate what I said in my response, which is essentially that there isn't really an easy way necessarily to evaluate the virus levels in the field, but you can take one of those pseudo-queen lures that I used in my experiment, and a beekeeper could put that on a frame when he or she is selecting attendants for shipping, and by just grabbing the bees that are attracted to the queen pheromone, and that would at least select for bees that are attracted to queen pheromone, and then could potentially be good retinue bees, and potentially have lower virus levels as a proxy.
Perfect. And it looks like someone had followed up with, do you have a hypothesis as to why the avoider bees avoid the queen?
Yeah, I mean, like I said, that the avoider bees have this higher level of black queen cell virus from my results. So, that's not necessarily that's a correlation that doesn't necessarily explain a mechanism for why they're avoiding the queen. But we do know that like I said, we do know that bees that are more responsive to queen pheromone generally have smaller ovaries. They have fewer ovarioles than bees that are less attracted to queen pheromone, and so. Although it hasn't been shown that ones that actively avoid the queen, what their ovaries look like, I would imagine that they probably have larger ovaries than bees that join the retinue. And exactly what's going on there, I'm not sure yet. It's sort of an open area for research.
Awesome. Looks like there's no new questions, so I'll go ahead to that other question that Austin you had answered. So someone had said they missed whether they used the data presented to include both native and honeybees, or whether you're still working on pulling together native bee data. Could you clarify and maybe expand on that a little bit if you have any expansion or thoughts?
Sure. Yeah, what I presented here today was my floral survey. And along with that, I'll integrate that into my bee data that I'm still processing. And that bee data is both in Philly and Montreal, and it includes all of the native wild bees and the honeybees and anything I found any bee species I found, including honeybees, will be a part of that. But as it pertains to honeybees, I wanted to highlight, especially bringing in that Sponsler study on the way that honeybees, in particular, according to the DNA analysis of their pollen, in urban landscapes, they really do rely on that spontaneous vegetation. I wanted to highlight that in this presentation today.
Perfect. And I guess the follow-up question was if you can share with the group how far away from you are you from publishing? Goodness.
I'm hoping, yeah, hopefully early next year. I'm hoping if I can keep on pace, I'll defend by the end of the year. Got all summer here. I won't need to be teaching over the summertime, so got all summer to speed through this, and then yeah. So as soon as I get those out, I see that Dewey's sharing an email there. I'll be sure to send those over, and hopefully that'll be early next year.
Perfect. And as a reminder, folks, if you have any questions that you didn't end up getting to ask during this session, you can feel free to always email the NAPPC email, or if you happen to jot down their emails from the presentations, feel free to email the different presenters directly. So I'll go over this question for you, Dr. Walton, again. I know you answered it, but someone had said, I found it interesting that the queen avoider bees had high viral load. Wait, no, I read that one. No, I didn't. Okay. I'm curious, do I know how these highly affected avoider bee approaches the queen? Why don't the retinue bees prevent the infected avoider bee from approaching the queen?
Yeah, well, for one, I mean, the other retinue bees may not, be able to sense that those bees have a higher level of viruses. So they may not actively prevent them from joining the retinue. But a lot of these, in the video I showed, that example, you do see a bee come in and approach the queen and then leave. But a lot of the avoider bees, it happens just as the queen is not actively laying an egg, but as she's moving through the colony, looking for a place to lay an egg, and she encounters all kinds of bees, and they behave in different ways. Some join the retinue, some briefly antenate her, return to work, some ignore her, it seems, but then there's others that will act, they'll encounter the queen and then they'll very quickly try to get, they'll move out of the way, and they will run even very far away, and so those are the ones that I count as these avoider bees. And so they're not necessarily going to the queen. The queen may just happen to be passing by them, and they make a point, I mean, that sounds subjective, but they clearly will avoid the queen. They will move away from her as soon as they encounter her. Those are ones that I consider avoiders.
Perfect. Reed, did you have a follow-up question?
No, I had a question for Chia, if that's okay. Okay. Yeah. I was just curious. So just wondering, I might have missed this in the presentation, but what about the mustard pollen is beneficial when looking at Nosema resistance. What about the pollen specifically is helpful in this case?
I think we're actually working on analyzing the concentration of glucosinolates in mustard. But from that study, we found in the 80s, the concentration is pretty low compared to what would have been found in like leaf tissues or like seed meals. Actually, mustard seed meal is what growers often buy as a soil fertilizer and in treatment. And I think that study in Europe, they actually fed mustard seed meals, which has a much higher concentration to bees than in pollen. So I feel, and another thing is, and I'm not sure if I mentioned that, even though the treatment is 5 grams of pollen or 10 grams of pollen we actually refill the pollen every other day. And then so there's 5 grams of pollen and 5 grams of the others, which is like goldenrod and asters and others, like, I don't know, fall legumes. I don't think that's what the bees, I don't think that's enough for what the colony needs, but we are confined in the state, in those like mesocosm cages. And the students were doing as much sorting as they can to provide that pollen. So that was probably another limitation. But I feel if we have the opportunity to give them more pollen, more mustard pollen, perhaps we could see some more benefits in terms of like a disease management.
Understood. Thank you so much.
Thank you.
Perfect. Next question is for Dr. Wagner from Dewey Karen. He said, do you think other pollicents or even pollen for forager bodies might also be groomed similar to powdered sugar?
Sorry, other what?
Like pollen or forager bodies might also be groomed to similar or to powdered sugar.
Yeah, it's a good point. I think bees typically do groom pollen, and that's one of the ways they move it from their body and the hairs on their body into the corbiculae. Yeah, I think so.
Perfect. And I know, Carrot, you had a question for Alexander. Do you mind sharing that question again? It was kind of in the panelists and host chat. I just wanted to give a chance for the audience to hear it as well.
Yeah, sure. I wasn't able to post in the Q&A for some reason. Let's see if I can go back and find it. Yeah, so I was wondering if the lower virus load in the retinue workers could be a result of the workers that are in the retinue being in the retinue and being around the queen rather than doing other activities during that time that could be more likely to expose them to viruses. So I was trying to remember exactly what age the retinue workers are and what other activities happen at that time. But for example, if they're feeding brood or doing something else that could expose them to a lot of other bees that may increase their chances of contracting viruses, maybe that could explain it. But Alex had a great answer. I can let you answer yourself.
Yeah, well, I mean, the retinue bees sort of span a long time. It's generally around 10 days is where I sort of witnessed like the highest level of that behavior, and so they are also doing nursing behavior. They aren't necessarily just staying in the retinue, so they should still potentially be interacting with brood, and of course they do spread the queen pheromone by interacting with other workers. So they should be actively trophallaxing with other workers and potentially getting viruses. But like I said, whether or not it's a passive or an active protection of the queen, even if it's a passive protection of the queen, then the result is still that the queen is protected from viral infection. But I was saying that I think my two findings, one, that bees that are actively avoiding the queen have higher levels of viruses. That seems like some evidence that it might be active. And then, of course, when I experimentally infect bees with virus and then I see that they have a lower response to queen pheromone, that also sort of suggests that there might be a potential active protection of the queen. But whether or not it's passive or active isn't totally known. I just think my results at least suggest a potential active protection of the queen.
Perfect. And I also wanted to make sure that I asked, do any of the other panelists have any questions for each other as well? You're also more than welcome to ask questions. Go ahead, Dr. Amiri.
Let me ask you a few questions that I have. I start with Chia. We know that mustard produces, there is a lot of secondary metabolites from the mustard pollen. Did you check whether there is any connection between those secondary metabolites with the Nosema reduction or so?
The main one, and that's the one that's most abundant in pollen, was sinigrin. And that one is at a much higher, I guess I'm not sure how much it is, but it's at a significantly higher concentration than there were a couple others. I can't remember the name, but sinigrin is the one that we're most interested in right now.
Because it seems that beekeepers are mixing mustard with sugar and feed it to the colony for that reason. So that's why I'm interested to see whether there is any connection.
Yeah, that would be very interesting. I would, the project kind of ended, and we currently not have upcoming plan to continue the mustard study at the time, but I would be very interested in the further investigate and see what we could do in terms of like laboratory experiment and also like field experiments about how to utilize this cover crop.
Right. Thanks.
Mhm.
And
Thank
then
you.
my next question is from Alex. Alex, why did you choose focusing on black quince virus for your field study? Why not other?
Yeah, well,
Viruses?
we're going to do a whole panel of viruses. We've just started processing some of these. So far, we've looked at a couple other viruses, and they've been, like IAPV, for example. They've just been at really low levels in our bees. So they've been we haven't really detected those yet, but we're in the process of we've extracted the RNA from all these bees, and we're just doing qPCRs now.
So
To
have you checked deforming viruses or sag brood virus or Lake Sinai virus to see how
Yeah, we looked at deformed wing virus. That one we didn't see a difference. But we haven't looked at sac brood yet, or
Lake Sinai virus?
Lake Sinai, or I don't think we've looked at
No.
cashmere virus yet. Yeah, we're
Oh, no,
just working
those
through
that
now.
are very prevalent in the colonies.
Yeah. So the one that so far that has had a significant difference has been black queen cell virus. So for my talk, I focused on the interesting results since there's only
And
10
then
minutes.
one graph that you showed from, well, both graphs, mostly the one that you showed about the one in the colony. How many bees did you collect from the colony, these retinue bees that you tested them and you found out
Yeah,
yeah.
right. Those retinue that figure that was around. I think there's a different sample size for each of those treatments because it's as many as will actually. That's an observation, so I can't control the sample size. But
Yeah.
it was like 10, 14. They weren't huge numbers, but we have more samples. I went in there multiple days, so those are all age matched at 9 days, I believe, and I went back in at like day 14. I went at day 10, and so there's more work that can be done just to check if the bees that join the retinue at these different ages, if they also follow that same pattern. Yeah. But I had to take a frame out of a colony and follow the queen, and sit outside and with the frame outside of the hive. And I generally did it for about an hour. And then after that, I didn't want to stress out the queen too much. I was doing it in the shade, trying to keep it cool, but I had to sit there and watch the frame and follow the queen. And I would only sample bees if they were painted, because the painted one, I painted them so I knew what age they were. So I wasn't grabbing just any retinue bee or any queen avoider. It had to be one whose age I knew. So, yeah.
Thank you. Do I have time to ask one more question?
I think one more question. Yeah, we'll have time for that.
Let me ask Kira. Kira, how do you see the method being used by beekeepers?
Yeah, well, my
Do
hope
you
is
think
to,
it's something that beekeepers need to use it, or like a specific type of beekeepers need to use it, or?
yeah, sure. My hope is to make something very easy for beekeepers that are interested in breeding or selection to use it. So I think, there's a lot of people are becoming more interested in social immune mechanisms of honeybees for defending against varroa and other diseases, especially in light of the growing resistance mites have to miticides that beekeepers have traditionally relied on. So I think, if we can come up with a really simple test that's feasible for beekeepers to do very quickly and very cheaply in the field, that it'll encourage more people to start looking for those traits and selecting for those traits. But first, this is just very preliminary. We have to first see if it even works and really correlates to the actual grooming, more complex and more detailed grooming mechanisms or grooming measurements.
If we review the literature, we see grooming behavior was in a focus on a focus for some years, and then people just kind of forgot about it, and then they tended toward hygienic behavior.
Mm-hmm.
So how do you know why? It's because of not having a well-established method to select grooming behavior in the colonies, or There
Yeah,
is
I think that's part of
connection
it.
between grooming and other health status, health traits in the colonies.
Yeah, I think that's part of it. I think grooming isn't, like a cure-all. You can still have high grooming bees that have pretty high mite loads. So I think it helps, but it's not like a silver bullet. Whereas hygiene, I think if you have very hygienic colonies, you tend to see lower mite loads. But I still think it helps. And I think anytime you simplify the process of a selection tool, you're going to get more people that adopt that selection tool. So there are still several groups that are selecting for those mite biters and groomers, but it's typically in the academic setting where people have funding and equipment and things like that. So my idea here was to try to make something that anyone can do in their backyard, whether they have 2 colonies or 10,000. So just trying to make the link between, collecting the data and accessibility for everyday beekeepers.
And then when you review the literature, you see even adding powdered sugar to the colony to encourage bees doing grooming was suggested by some people, but by the end they said it's not efficient.
Yeah, I think I'm not sure that's an efficient method for controlling varroa. And we're not going for actually inducing grooming for the purpose of controlling varroa, more inducing grooming for the purpose of measuring how good the bees from that colony are at performing grooming in general.
Yeah.
If that makes sense.
Yeah. Thank you.
Well, thank you all for all of your questions and all of your research that you all have done this year. If folks have any follow-up questions that they have for any of the presenters, again, you can email me at nappc@pollinator.org. And I'll make sure that I'll follow up with those questions with each presenter. Thank you for all of our sponsors, our presenters, our researcher, our task force co-chairs who've been on the call and helping out with this whole process. If you all are available next week, our Bombus and Lepidoptera Bombus and Lepidoptera virtual reporting webinar is next week. So you'll be able to actually join that. It's around the same time. Next week, I believe it's 2 p.m. Eastern to 4 p.m. Eastern time. So feel free to join us for that. Again, if you have any follow-up questions, feel free to email me and I will make sure that someone is able to follow up with you. Have a great rest of your day, everyone. Thank you so much for joining us. Bye, everyone.
Thank you.
Thank you.